Before antibiotics, a strep throat could become a fatal infection. A cut that became infected could kill you. Childbirth was dangerous partly because puerperal fever, caused by streptococcal infection, routinely killed new mothers. The discovery of penicillin by Alexander Fleming in 1928 and its clinical development in the 1940s fundamentally transformed medicine. Bacterial infections that had been virtually untreatable became curable within days. Life expectancy in the developed world rose sharply, and modern surgery, which depends on the ability to control postoperative infection, became possible. Today, antibiotics are among the most prescribed drugs in the world. And that ubiquity, combined with misuse and overuse, has created one of the most serious threats in contemporary medicine: antimicrobial resistance. Antibiotics work by exploiting molecular differences between bacterial cells and human cells. Because bacteria are prokaryotes, lacking a nucleus and having fundamentally different cellular machinery from our own eukaryotic cells, they have structures and processes that we do not, and those differences are the targets. Beta-lactam antibiotics, the family that includes penicillins and cephalosporins, inhibit the synthesis of the bacterial cell wall by binding to proteins called penicillin-binding proteins, or PBPs. Bacteria have a rigid cell wall made of a mesh-like polymer called peptidoglycan that maintains their shape and resists osmotic pressure. Without PBP function, the cell wall cannot be assembled or repaired during bacterial growth, and the bacterium lyses, essentially bursts, under its own osmotic pressure. Human cells have no cell wall and no PBPs, so beta-lactams have no equivalent target in our cells. Different antibiotic classes target different bacterial vulnerabilities. Fluoroquinolones, including ciprofloxacin, target DNA gyrase and topoisomerase IV, enzymes that bacteria use to manage the topology of their DNA during replication and transcription. Inhibiting these enzymes causes lethal DNA strand breaks throughout the bacterial chromosome. Macrolides, including azithromycin, bind to the 50S subunit of the bacterial ribosome and block the elongation of the protein chain being synthesized. Tetracyclines bind to the 30S ribosomal subunit and prevent aminoacyl tRNA from entering the ribosome's A site, blocking the addition of new amino acids. Aminoglycosides also target the 30S subunit but cause misreading of the genetic code, producing non-functional or toxic proteins. The specificity of these mechanisms, bacterial ribosomes differ structurally from eukaryotic ribosomes, is what allows antibiotics to kill bacteria without killing human cells. Bacteria have evolved resistance to antibiotics through several distinct mechanisms. Beta-lactamases are enzymes that bacteria produce to chemically degrade beta-lactam antibiotics before they can act, cleaving the beta-lactam ring that is essential for the drug's activity. Extended-spectrum beta-lactamases, known as ESBLs, can inactivate a broad range of beta-lactams. Efflux pumps are membrane proteins that actively pump the antibiotic out of the bacterial cell faster than it can accumulate to a therapeutic concentration. Target mutation changes the molecular shape of the antibiotic's target. For example, mutations in penicillin-binding proteins reduce the affinity of beta-lactams, the mechanism behind methicillin-resistant Staphylococcus aureus, or MRSA. Enzymatic inactivation by transferase enzymes modifies the chemical structure of aminoglycosides, rendering them inactive. These mechanisms can spread between bacteria not just through reproduction but through horizontal gene transfer, bacteria sharing resistance genes across species boundaries through plasmids. The World Health Organization has identified a list of priority pathogens, bacteria for which new antibiotics are most urgently needed, including carbapenem-resistant Enterobacteriaceae, Acinetobacter baumannii, and Pseudomonas aeruginosa, all of which have developed resistance to nearly all available antibiotics. Currently, antimicrobial resistance is estimated to directly cause approximately 700,000 deaths per year globally. Without intervention, projections suggest this figure could reach 10 million deaths per year by 2050, surpassing cancer as a cause of mortality. The economic and medical consequences of entering a post-antibiotic era, in which routine surgery, cancer chemotherapy, organ transplantation, and neonatal care become far more dangerous, constitute one of the most serious threats to global health in the coming century. Antibiotic use also disrupts the gut microbiome, the trillions of beneficial bacteria that normally inhabit the intestinal tract and perform critical functions including vitamin production, immune system development, and colonization resistance against pathogens. When antibiotics deplete the normal microbiome, the ecological niche it occupies can be colonized by opportunistic pathogens. The most important example is Clostridioides difficile, C. diff, a spore-forming bacterium that causes severe colitis and diarrhea when the normal gut flora is depleted by antibiotic treatment. Antibiotic stewardship, the systematic effort to optimize antibiotic prescribing, using the narrowest spectrum appropriate drug for the shortest effective course, is both a clinical practice and a public health imperative. Every unnecessary antibiotic prescription is a small contribution to the global resistance crisis that will define medicine in the next generation.